Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT class 12th molecular basis of inheritance, that is part 6. Next we discuss genetic code. During the replication and transcription, a nucleic acid was copied from another nucleic acid. So here one nucleic acid was copied from another nucleic acid. For example, here they have one nucleic acid. Here when you take as in here one nucleic acid, one nucleic acid to the information to get passed into as another nucleic acid. This total process is called as what? That is in a genetic code. That is called as what? Completely in that genetic code. Hence, this the process are easy to their conceptualize on the basis of any their complementary. Means how does that takes place? Means one strand, the two strands they get had as in a the anti-parallelly. This information to get copied from as in here to here, and the process of the translation is requires transfer of the genetic information from the polymer of nucleotide to the synthesis of as any polymer of amino acid. So here the simple saying as what? Here the information to get passed into as any their polymer of the nucleotide to as any polymer of that is what? Amino acid. Polymer of amino acid. And neither does any the complementary exist between the nucleotides and amino acids, or not could be they are drawn in theoretically. So before when we are observed to as in theoretically in this total how to the genetic code will be there takes place. Now once we observe, okay, let's there existed the ample that evidence through as in the to support the notion that the change in the nucleic acid means the genetic material where they are responsible for the exchange of amino acid in a protein. So here what happened? Here the mostly the one nucleotide will be the get change. What happened? Here amino acid base also automatically to get change. Clear? And this lead to as in the proportion of the genetic code that could be the direct the sequence of amino acid during the synthesis of a protein. So here amino acids will be to get how to the synthesize with help of the nucleic acid to get released as means they are provided as in some amino acids. That amino acid they are completely to get participate in the DNA protein synthesis. Okay, that is. And here the determining next let's if determining the biochemical nature of the genetic material and the structure of DNA was the existing the proportion of any the disappearing of the genetic code were the most challenging. So after what is in a biochemical where which one they are complete they get change of genetic information from one generation to other generation, which one they are complete they get transfer from generation to generation, that they're mostly the challenging process in for those days. Here after very the true sense the required the involvement of the scientists, the several their disciplines, the physiologists and their organic chemist and biochemist and the genetics. Means here the different like as any the genetics scientist and physics scientist and chemistry scientist, like the different scientists did as in that experiments on the DNA. Means the which they're carrying from the information for as in one generation to other generation. They're excited they are did as a number of experiments on the DNA. Okay, let's see that first one. It was in the George Gamow. It was in the physicist who was argued with as in the science. That argued that as in since they are in only the 4 bases and they are having the code for as in 20 amino acid. So for example, when we are observing as in here 4 bases as present. Here how many bases? They having as in 4 bases as present. In this completely 4 bases, they're bind with as how many amino acids? They're bind with as in they're completely in a 20 amino acids. They bind with as in a 20 amino acids. Who the said? The scientist was in a George Gamow. Okay. Next, the code should be their constitute the combination basis. How their basis? For example, when you take as any that combination of the any basis, the basis will be they having as in the completely therefore. Like when you observe to as any the uracil, cytosine, adenine, like these are complete the 4 bases will be the complete they get formed. Okay, and they suggested to as in that in order to code as in 20 amino acids, that code should be there made up of as in 3 nucleotides. They're made up of what? Each one they having as in completely their 3 codons will be they get present. There are 3 nucleotides as present. Like when this was in very there but proportion because they are in a completely the primation the combination they're given as in simply 4 cube. That is what 4 cube means. What here they get as in a 4 into 4 into 4. Here they complete get as how many? That 64 codons will be get. How many? 64 codons would be there completely. They generate 64 codons. Generating maybe there are more codons they are required. So here in this total their genetic information, how do they copying in this total that in 61 codons they get participate to bind with as in a specific amino acid. Here for example I am taking as in a 1 codon. That codon I am taking here the AUG. It is in a 1 codon. This AUG to compulsory they are bind with as in a 1 specific amino acid. They bind with as in a 1 particular specific amino acid. What I am saying now? Yeah, I am saying only one thing. Here 61 codons to get bind with as in a specific amino acid. Only 3 codons does not code for any amino acid. That will be called as what? That comes next to that is the stop codons. Am I right? Let's see here. Here the providing the proof of that in a codons was in it the triplets. Triplets means what? They given already here. See, that is in AUG. Adenine, uracil, and guanine. Here, how many the amino acids are? How many codons is present? They having a triplets. It was in more there that during that task in the chemical method as developed as in Hargobind Khorana. The scientist was whom? The scientist Hargobind Khorana. He also there how to the RNA synthesis was takes place. He was clearly there was explained. See here biosynthesis of in RNA. See here he explained about as in the instrumental the synthesizing mRNA molecule define the combination of the bases. That is what homopolymers and copolymers. Very very important. Here Hargobind Khorana he was there mostly did as an experiments. They are instrumental there. Very very important. This is see here the instrumental in the synthesis of RNA molecule. Synthesis of what? That is in a RNA molecule define the combination of in a homopolymers as well as in a copolymers. Okay, and after the next the scientist was whom? Their Marshall Nirenberg. He was explained about as what the completely cell-free system. He explained about as what? In a cell-free system he explained. Cell-free system for as in a protein synthesis finally they help to as code for as in they are in a disappear. So each codon they bind with as in that particular amino acid he was there explained. And next the several oxa enzyme that is in a polynucleotide in a the phosphorylase was helpful to as in the polymerizing of RNA with defining the sequence, the template and independent manner process. So for example when you take as in one particular strand of the DNA, am I right? In this DNA where they completely they get act as a template strand. They get act as in the template strand. So here the servo-ocasa enzymes they are completely participate in the enzyme. The enzyme they are completely participate in the polynucleotide their phosphopolymerase enzyme to get participate. Here they completely they bind with as in the specific amino acid into as in a specific nucleotide. Like next, see here what is the function of the enzyme? It is get act as an enzyme synthesis of any RNA. Already you know that one. What is that enzyme? DNA dependent RNA polymerase enzyme that could participate in the DNA formation of the template. Clear? That is next. Here finally in the checkerboard they given up for as a genetic code was there prepared which as in the given time that table 5.1. So in the scientist how do they easily to get identified then in different codons they given as a one Punnett square in a one checkerboard type. Like once you observe here the time that table also they given here. Here it is in a first position, it is in a second position, it is in a third position. Okay, see in that first position he taken as any uracil, cytosine, adenine and guanine. See these are the incompletely uracil, cytosine, adenine, and guanine. And second position also uracil, here cytosine, adenine, guanine. And in the third position, where you notice, see here we make compulsory as in the 4 codons, that is in uracil, cytosine, adenine, guanine. Here also the same 4 codons to get they mentioned. Here 4 codons here completely to get mentioned. Let us see here once here in these three uracil, 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 cytosine. Like you completely they are make as in a we get as in this total the Punnett square will be the comes in the different amino acids. Here each one to get bind with as in these 4 codons. In this completely cytosine, uracil, uracil, cytosine, uracil, cytosine. Like this total we are comes as in this total Punnett square. Okay. Like here total how many amino acids that complete the different the 64 amino acids comes in that in they completely 61 only they are complete the code for as in a particular amino acid, and then 3 are not code for as any amino acid. Okay na, these are complete amino acid like as phenyl alanine, leucine, isoleucine, methionine, valine, serine, proline, tyrosine, alanine, tryptophan. These are the C. Here they given as in STOP means stop codons. Before I say what I say only in A the complete 61 codons their code. 3 codons does not code. That code completely called as what? Stop codons. That codons that does not their bind with as any amino acid. Okay na? This is in a completely in a the genetic code map. Clear ma? This is next. So they given some features also here. Let's see one by one. That features the silent features of the genetic code as follows. The first one, the codon is triplet. Already I said that one only. What is that? The AUG. Here the AUG, see it is in a triplets. How many is there? 1, 2, 3 triplet, and 61 codons code for as an amino acids, and 3 codons do not code for any amino acid, that we call as an stop codons. 61 codons only to get bind. 3 is not code for as any amino acid. Correct? This is the first one. And second one, some amino acids code more than one codon. That code will be that degenerated. For example, here AUG is there. If in case we get add with as any the cytosine, it is a bind. It's not possible because only the particular more than one codon to get bind. What happen? Amino acids complete they get degenerated. Next, the codon is read for as mRNA. The continuous fashion. There are no punctuations. See, it is continuously they're going on. The continuously the code will be they get synthesizing. They have no any gaps here. Any their breaking is not takes place. Once they get stored in a reaction, it is a continuously will be they get takes place. Okay, and next the code nearly universal. The example for the bacteria, human, the completely UUU. It is code for as in a phenylalanine. That is in a some the exceptions of this NA rule have been found in the mitochondrial codons and in some protozoans. So mostly in that codon they have it universally, means some they having as in a some groups they having in the same all codons will be the same. For example, they given here that is what phenylalanine, that is in a UUU. It is mostly be seen which members like as in a in some mitochondrial codons as well as in some protozoans also they contain this kind of the universal codons will be they get present. Okay. Next one is see here the AUG. AUG there act as any the dual function. That is what their methionine. AUG means what? That is act as a dual function. Its code for as a very important, that is a methionine. Right? And it is also act as in the initiator codon. The mostly here the AUG they get act as any initiator codon. Okay. Let us the UAA, UAG, UGA. These are completely called as any their stop termination codon. Means simply called as what? They are called as stop codons. They called as what? In a stop codons. Let's see here. The following the sequence of nucleotide mRNA predict the sequence of amino acid the code for as and take help of in a checkerboard. So already you know here AUG. AUG means what? It is a part of the completely what ma say it is in a methionine. Let's see here. See AUG means what? Where is that? See here AUG means what? It is a completely methionine. It is what? In completely methionine. And next to here they given as what? It is in a UUU. UUU means what? That is in a completely phenylalanine. UUC means there is a phenylalanine. Like these are completely called as what? They given as a one by one in particular amino acid. Next, here now write the opposite. Methionine means what? That is completely called as an AUG. Phenylalanine means what? UUU. Like that, enter the codons we did get right as usually, like as in a this pattern. Okay, now they given here right. To discuss in next class. Thank you for watching. Please share, subscribe, and like.